mRNA, which is essential for translating genetic information from DNA into proteins, is a key molecule in disease research and the design of mRNA vaccines and drugs. For accurate functional analysis and safe applications, obtaining high-purity mRNA with intact structure is essential. In fact, central research in modern biotechnology, such as disease research, drug development, and vaccine design, focuses on the expression and regulation of mRNA. mRNA is also used as a diagnostic biomarker and a key molecule for therapeutic development. For this reason, pure and precise mRNA extraction technology is the starting point of all life science research and the foundation for next-generation bioinnovation. Thus, mRNA is a key component of life science research, but it is a very unstable molecule that is easily degraded. Therefore, to obtain reliable and precise research results, it is vital to secure pure, high-quality mRNA. Among total RNA, mRNA accounts for only 1 to 5%. Genomic DNA contamination during analysis can create fatal noise. Additionally, DNase I treatment during extraction not only increases costs, but can also result in the loss of precious mRNA samples during the process. With just the EcoQprep mRNA kit and magnetic separation rack, you can quickly extract high-purity mRNA from any sample, cells, animal, or plant tissues, without DNase I treatment. The EcoQprep mRNA kit uses Oligo DT magnetic bead technology to remove unwanted rRNA and cDNA and rapidly and precisely isolate only mRNA. It offers over 70% faster extraction time compared to conventional column-based methods, finishing in just 10 minutes what would normally take 40. EcoQprep instantly overcomes the time and efficiency limitations of other magnetic bead-based kits. Along with fast extraction, it delivers powerful performance, offering high sensitivity to detect even low-expressed genes clearly and ensuring precise quantification, reducing both research costs and time. It is also fully compatible with the EcoQprep 96 Lite automation system, allowing up to 96 samples to be processed simultaneously in just 12 minutes. By exchanging the magnetic rod, you can also extract up to 24 samples in MIDI scale, offering solutions optimized for various experimental scales. EcoQprep is more than just an extraction kit. It's a solution that combines reliability, efficiency, automation, and cost-effectiveness. Reclaim the golden time of your research. Fast, accurate, and in a class of its own in efficiency. EcoQprep will elevate your research to the next level.